Hello, my name is Trina Ganguly and the date today is February 21st, 2023. Today I will be discussing three different types of immunological assays. First, I'll begin with fluoroimmunoassays. In this image, we can see the procedure for this type of immunological assay. Fluorescence immunoassay is a simple, rapid, and sensitive technique that is used to measure many compounds, including drugs, hormones, and proteins. This method has been widely applied in the in vitro diagnostics industry, and in a classical FIA, fluorescent dyes that illuminate in UV light are used to detect the specific binding of an antigen and antibody. Also, a modern fluorescent-based immunoassay uses the detection reagent, a fluorescent compound which absorbs light or energy, also known as excitation energy, at a specific wavelength and then emits light or energy at a different wavelength. In this assay, the antibodies are labeled with a fluorescent molecule, and so once they have bound to the target antigen, the fluorescence label can be quantified, which indicates the concentration of the antigen. FIA can be used for the characterization of the affinity of specific IgG antibodies as a diagnostic test in various clinical laboratories. This type of immunoassay can also be used for the detection of a wide range of biomarkers to assist with the diagnosis and monitoring of several medical conditions, including cardiovascular disease, inflammation, kidney disease, thyroid conditions, and also including fertility. Next, I'll be discussing the radioimmunoassays, and so in this image we can see what to expect from using this type of immunoassay. Radioimmunoassays have many advantages when they're compared with other analytical methods, such as high specificity and high sensibility. And so, as a result, it can be used to determine all kinds of trace substances with immune activity. RIA is a sensitive and quantitative method that was first developed during the 1950s. This method is used to determine the concentration of the substance, and it usually uses antibodies to measure antigen concentrations, such as the hormone levels within your blood. Although the cost of this technique is very low, this method is also very specific and is known to be very sensitive. And currently it is widely used within the IBD area. RIA is also considered to be the gold standard method when it comes to determining thyroid hormone levels, mainly because RIA exhibits high sensitivity and specificity and low detection limits. In human beings, RIA of the TSH hormone is routinely performed to assess hyperthyroidism and hypothyroidism. And in terms of how it works, ABS or AGS is labeled with a radioactive isotope, which can be used to detect minute amounts of a corresponding AG or AB. And it then compares the amount of radioactivity before and after the incubation period within a known labeled AB or AG. And lastly, I'll be discussing ELISAs, which are enzyme-linked immunosorbent assay. And in this image, we can see how this type of assay works. Enzyme-linked immunosorbent assays, or ELISAs, can be used to detect antigens, antibodies, and haptens. It is a highly sensitive tool within in vitro diagnostics, or IVD. ELISA is based on the concept of antigen-antibody reactions, and it can directly determine soluble antigens or antibodies within bodily fluids. In real-world applications, it can be used for clinical diagnosis, disease surveillance, disease census, forensics examinations, various veterinary purposes, and also agricultural plant disease diagnosis and verification. And ELISA, similar to other types of immunoassays, also relies on antibodies to detect a target antigen using highly specific antibody and antigen interactions. In an ELISA assay, the antigen is usually immobilized to a solid surface, and this is done either directly or through the use of a capture antibody itself being immobilized on the surface. An ELISA test uses various specialized enzymes that attach to antibodies in your blood, and so as a result, this test involves mixing a sample of your blood with a known compound on special absorbent plates. And depending on what your doctor is diagnosing, the test can use various types of enzymes in order to identify the various types of antibodies that can be tested for. And so in conclusion, overall the immunoassay testing works by using antibodies to detect small biological substances within the blood and other bodily fluids. And the method follows the aspect that particular antigens bind to specific newly introduced antibodies, and as a result stimulating an immune response. There are 5 types of overall immunoassays, which are the most commonly used types, and they are enzyme immunoassays, radioimmunoassays, fluoroimmunoassays, chemiluminescent immunoassays, and counting immunoassays. These immunoassays are methods for detecting and quantifying analytes within a biological sample based on specific antigen-antibody reactions, and they can be used to detect proteins, lipids, nucleic acids, or small molecule targets, as long as there are known antibodies that bind specifically to that target. And depending on the type of immunoassay you are using, they will detect different types of molecules using the various different types of methods within each of the types of the immunoassays discussed. And here are all my references that I used for my presentation. Thank you.